Hello, everybody. Thank you very much for joining us today. My name is Gabrielle Richardson, and on behalf of Megan, I'd like to give you a warm welcome to this Megan Masterclass with our guest speaker, Professor Rik Henson. Just before we start, and whilst people are still joining us, we have a few housekeeping rules to go over. Please use the Q&A button on your screen to send in your questions at any time during the masterclass. There will be time for Professor Henson to answer questions you may have at the end of the session. Any questions unanswered, we will try and respond in writing after the session. I'd like to take this opportunity to remind you that this webinar is being recorded with the permission of our speaker, and we will be making it available to watch on demand after the session today. And finally, just a reminder to please adhere to the MEG Code of Conduct for webinars and engage respectfully online. So without further ado, I'd like to kick this off, and it gives me great pleasure to hand over to Professor Henson, who will start the webinar. Hello, Rik. Good morning.
Hello.
We'll start the webinar now.
So I'm going to talk about the use of MEG to study healthy and unhealthy aging. As I'm sure you're aware, people are living longer, which is a good thing, of course, but the proportion of the world's populations that are in old age or in retirement is increasing, which imposes societal burdens. And we know that aging does bring, unfortunately, cognitive problems owing to brain changes. So understanding how aging of the brain affects cognition might help us maintain cognitive abilities for longer in old age and help us to function independently for longer. Neurodegenerative diseases such as Alzheimer's cause cognitive problems, that is the definition of dementia, and age is a big risk factor. So I think 1 in 3 people over 80 go on to develop Alzheimer's disease, bringing huge individual and, again, societal burdens. So understanding how such diseases affect the brain and cognition may help develop and also monitor the efficacy of pharmacological treatments, for example. Now, MEG is one of several brain imaging techniques that we can use to study the brain and relate the brain to cognitive abilities, but it has unique advantages over others like MRI. And that's what I hope to focus on in this talk. Firstly, a bit about the aging brain. Well, this is a transverse or horizontal section through the brain of a 19-year-old with the grey matter around the edge and the lighter white matter. It's a T1 weighted MRI image. And in a moment, I'll show you brains of people as they get progressively older, up to 88. Unfortunately, MRI hasn't been around long enough to track the same person's brain across so many decades. So these are all different people, but we've roughly morphed their brains into the same shape. You can still see, if you look closely now, gradual thinning of the grey matter around the edge, a reduction in the white matter, and as a consequence, the ventricles, which these darker regions in the middle, tend to get enlarged in old age. So if I now show you the 86-year-old on the right and the 19-year-old again on the left, you can see what dramatic effects aging has on the structure of the brain. These images are from the CamCAN dataset, which I'll talk about later. We also know that age affects our cognitive abilities, at least some of them. So what we call crystallized intelligence, or language, world knowledge, etc., doesn't tend to decline much, whereas fluid intelligence, the ability to solve novel problems, does decline dramatically, as does, as we often experience, our memory as well, or episodic memory. So at least some types of cognition do change dramatically with age. Here's now longitudinal data. So these lines join the same person, lots of different people, and 2 observations on the same person. This is the volume of the hippocampus, which is a structure known to be important for memory. On the left is the same people, but now we're measuring their memory ability. And you can see there's a pretty uniform decline in hippocampal volume on the right. On the left, there are some individuals who show quite a dramatic decline in their memory in their old age. There's others who appear to maintain this. Now, some of this may just be measurement noise, but in general, we see this pattern a lot, where some people can maintain their cognitive abilities despite the structural brain changes I showed you earlier. This is sometimes called cognitive reserve. But it suggests to me that there are other things beyond brain structure, or brain volume measured by MRI at least, such as functional activity or functional connectivity or reorganization or compensation that explain this more complex relationship between structural brain changes and cognitive ability. So I've been showing you MRI scans, which has been used for many years to study aging. But of course, structural MRI is a static measure of the anatomy at one time point. Whereas functional MRI is a ability to track neuroactivity or connectivity during a particular task in the scanner over a matter of minutes. And fMRI has also been used to study aging, but we have to remember that the fMRI response is a function of both neural and hemodynamic properties. So it's affected by the vasculature of the brain. And we know that aging affects both of these. So we're interested in the effect of age on neural activity primarily, but we know it also affects hemodynamic vasculature, or the plumbing of the brain, if you like, which affects the bold response. So we need to disentangle these two. And furthermore, with fMRI, the hemodynamics are very slow. We're taking a brain image every second or so, and the hemodynamic response itself is very sluggish. So it doesn't peak until about 5 to 6 seconds after neural activity and can last up to 20 or 30 seconds. MEG, on the other hand, is a more direct measure of neural activity, much like EEG. So it bypasses any vascular effects of aging. And we can basically sample brain activity as fast as we want, so with millisecond resolution. And this opens a window on a rich repertoire of brain oscillatory activity, different rhythms that you might have heard about, alpha, beta, gamma, etc., which are all above kind of the limit of fMRI. The limit fMRI could measure about 0.1 rhythms of 0.1 hertz, but we're talking about brain activities of 10 to 100 hertz. You're probably familiar with EEG, which has been around for a long time. The main advantage of MEG is the fact that the skull and scalp distort magnetic fields less than they do electrical fields, and therefore MEG has a greater spatial resolution than EEG, or it can resolve more nodes in a network, or more states of the brain, spatially distinct states, which I'll come back to. Right, so today I'll start with healthy aging and introduce you to the CamCAN dataset. I'll talk about the effects of age on functional activity, and demonstrate what I said earlier, that there is an important vascular component to fMRI, and then talk about small latency changes in evoked responses recorded with MEG that would be invisible to fMRI. I'll then turn to functional connectivity, talk about brief microstates that last a few hundred milliseconds, measured with hidden Markov models of MEG data, and talk about a property of networks called functional segregation, which is believed to be important for cognition and to change in aging. In the second part, I'll turn to unhealthy aging, or dementia. I'll talk about a different dataset, the BioFIND dataset. Talk about some recent work using machine learning, where we've argued that MEG provides additional information beyond MRI, so it might be a useful biomarker for early detection of Alzheimer's disease. And I'll finish with what I think is the future of MEG, which is actually constructing neurophysiological models that relate parameters like neurotransmitter concentrations that we know are affected by drugs, right through to the magnetic or electrical activity in cells recorded by MEG. So starting with CamCAN. CamCAN stands for the Cambridge Centre of Aging and Neuroscience. It started in 2010. Here's a website if you want to read more about it. But in 2010, we knocked the doors of nearly 3,000 people in the Cambridge area, and they had to opt out. So this is more population representative than many other samples. It's not perfect, but we had about a 35% compliance rate, which is quite high. And then they did 2 hours of home interview, which included details about their lifestyle, demographics, etc. Then we got 100 of these people, a subsample, 100 per decade, from 18 to 88 years of age, from that group, and that subsample matched for sex, came in to the lab several times to do in total about 7 hours of cognitive tests, so very deep cognitive phenotyping. an hour of MRI, and today I'm only going to focus on the fMRI mainly, with a bit of diffusion imaging. And importantly, half an hour of MEG, which I'll talk about later. In 2016, we made the data available on request, subject to certain constraints. And we've had over 1,000 downloads and close to 100 publications. And again, if you want to access the data, the link is here. If you would click that link, you would enter our data access portal. You can see that it's been widely used. You have to agree to certain conditions, a data usage agreement, and provide your details, etc. But then you can select any type of data you want, MRI and or MEG, and here the MEG is offered at different stages of pre-processing. So you can also choose certain denoised versions, etc. Anyway, using CamCAN, one of the first studies we did looked at sensory evoked responses. So this paradigm, which we did in fMRI and in MEG, involved visual checkerboards, left and right fixation, concurrent with auditory tones to left and right ear. And when they heard this, they pressed a right finger key, key with their right finger. So this is a sort of sensory motor task. And a paper led by Karman Zetvanov looked at the vascular components of the fMRI response. So if you do a standard analysis, voxel-wise analysis, looking for the effect of age, so here any area that's blue shows a decreased sensory evoked response with aging. Red is an increase. This is correct for more comparisons across the brain. And this is I call this unscaled by RSFA because this is from standard analysis. And you can see that the visual cortex at the back here and the auditory cortex both show reduced responses or reduced fMRI signal in response to the visual auditory stimulus as people get older. And that's been reported many times before. But how much of that is due to the aging vascular system and how much is actually decreases in neural activity? Well, one way to address that is to scale the data by something called RSFA, resting state fluctuation amplitudes, which I'll come back to in a moment, but you can think of it as a proxy or a surrogate for vascular reactivity. And when we adjust for this, a lot of these age-related decreases disappear. So if you focus on the visual cortex here, the blue dots are the standard analysis where there's a significant decrease with age. But once we adjust for RSFA, there's no longer effective age in this visual region. The same is true for auditory cortex. It doesn't remove everything, by the way, because this ipsilateral motor cortex, so right motor cortex because they respond to their right hand, so the ipsilateral motor cortex actually shows age-related increases in activity, an interesting phenomenon in its own right, but that is unaffected, or in fact gets more significant in terms of R squared following scaling by RSFA. So it doesn't wash out everything. But nonetheless, we have to be careful with these scaling techniques because it's possible that RSFA isn't a pure measure of vascular health, but also does include a neural component. So we can actually test this though using MEG. So what we did was a mediation analysis. So we're asking how much of the variance in RSFA is caused by age, and of that variance, how much is mediated by various factors. If we included various measures of vascular health, so we also have blood pressure, we can look at heart rate variability from ECG or pulse oximeter, etc. A principal component analysis of these vascular factors showed a significant mediation effect. In other words, the effect of age on RSFA was diminished when we take into account vascular effects. And that suggests that vascular effects at least partially mediate the effect of age on RSFA. That's consistent with RSFA reflecting a vascular measure. But we can also ask, is there a neural mediator? And this is where the MEG came in useful. And we use a wide range of MEG measures, power, etc. And the bottom line is that these didn't show any significant mediation effect. So the effect of RSFA appears to be independent somewhat of the effect of age on neural activity. And this suggests that RSFA scaling is dominated by vascular components and therefore a valid way to correct fMRI data for age-related vascular changes. Just say that all again, conventional fMRI analysis would lead to the conclusion that older people have less neural activity in visual or auditory early sensory regions. But when you adjust for age-related changes in vascular health, there are no longer any significant effects of age that can be captured by fMRI. And MEG supports this use of RSFA because no MEG measure of neural activity mediated the effect of age on RSFA, unlike the vascular measures which did. But I'm not trying to claim that age has no effect on neuroactivity in early sensory regions, just that it's hard to detect those with fMRI. Because maybe age does affect neuroactivity, for example, the latency of evoked responses, that's at a scale which you just can't see with fMRI. And that brings me to this second study, which used the same task, but now in MEG. And this was led by Darren Price, a postdoc on the CamCAN project. So Darren looked at, first he estimated the sources of the visual and auditory evoked responses. And this is the topography over the MEG sensors. And this is the source reconstructed activity in the visual cortex using what's called multiple sparse priors. And we can look at sort of the average response across all people, showing a typical biphasic evoked response. But we can also look at individuals here. So now we're looking from above, if you like, where red means increases and blue means decreases. And each subject is a separate row of this matrix, which is ordered by age. And what you can see is as people got older, their responses got later and later. But in fact, this appeared to be a fairly constant shift. So the response just seemed to onset a bit later for the visual response. We also looked at the auditory response. Again, if we extract activity in the auditory cortex, we get a slightly different morphology of the waveform, but again, plotting against age, now we see a slightly different pattern. Here, as people got older, there seemed to be a stretching or dispersion of the response rather than a constant shift like the visual evoked response. There's now a more dispersed and delayed response in the auditory cortex. That's obviously just visualization, but to test it formally, we basically extracted using principal component analysis sort of the average temporal waveform. And then for each subject's visual auditory cortical responses, we fit them using that template, but we changed both its amplitude and scaling for different subjects. And importantly, we varied two delay parameters, a constant shift, left, right, if you like, of each individual's evoked response to fit the average, and a cumulative stretching of time where the delay got larger further away from time zero of the onset of the stimulus. So we fit this for every subject. We get an estimate of constant delay, which is the left column here, and cumulative delay, which is the right column, for both visual and auditory stimuli, the top and bottom row. What you can see is there's a significant effect of age on the visual constant delay and on the auditory cumulative delay, but no effect of age on the cumulative delay for the visual or the constant delay for auditory. So it appears that age has different effects on different sensory regions. In fact, that's supported by the fact that the correlation between the two delay parameters for auditory and visual was surprisingly low once we adjust for age. So they're almost independent effects. We went one step further and asked, what could be causing, or at least mediating, this effects of age on latency? And I showed you a mediation analysis earlier for RSFA. Here we're doing a mediation analysis for every single voxel in the brain, where the mediator, in this first case, is a measure of white matter integrity. It's called mean cortices, but the point is it measures white matter integrity. So these red areas here were areas that, white matter areas that mediated the effect of age on the visual constant delay. And they look like the optic radiations, so consistent with a delayed transmission of information to visual cortex. We didn't find any effects of grey matter as a mediator on the visual offset delay. We did the same for the auditory cumulative delay, and here there were no effects of white matter, but now there was effect of grey matter volume, at least in the auditory cortex here. So again, different possible structural correlates of these delay effects. So age exerts differential and uncorrelated effects on visual evoked latency, or constant delay, and auditory evoked latency, which is a cumulative delay. White matter seems to mediate the visual effect, consistent with delayed transmission of information with aging, whereas grey matter volume seemed to mediate the effects of age on the auditory cumulative delay, which we can speculate might reflect sort of delayed local computation in the neurons in that part of the brain. But the bottom line is that MEG reveals multiple contributions to age-related neural slowing. Turning now to connectivity and this concept of microstates. This was stimulated by a paper from Oxford several years ago now, where they used hidden Markov models to decompose MEG data into a series of discrete states with distinct spatial patterns of connectivity. So with the hidden Markov model, the brain is only in one state at a time, and this is time left to right here. And here are 3 example states, different colors. So you might be in a blue state, then switch to a red state for a short time, then go to a green state for longer. So these time courses are estimated from the model, and each of these different colors or states is associated with a particular covariance pattern over the sources of the amplitude envelope. So these are like brief, and these are the time's not shown here, but it's usually typically hundreds of milliseconds. So we think the brain cycles through discrete states quite quickly at rest, and we can summarize the temporal properties of these states with a number of measures, such as how much of the total time is spent in that state, the mean lifetime of that state, a number of occurrences, the mean interval length, etc. These are correlated measures, but they give some summary of the temporal dynamics of these microstates. So use this method, Ronnie T-Bon and us looked at the effect of aging on CamCAN's resting state MEG data. So now we're talking about resting state MEG data rather than the task data. We had about 600 people who had clean MEG data of about 8 minutes. And because we have multiple measures of these brain states, so if we have, say, 8 brain states, each with these 4 temporal properties, we have 32 features from the brain. And we also have a dozen or so cognitive measures, which are all correlated. So we use something called canonical correlation analysis to look for linear combinations of brain measures, microstate measures, that relate to certain linear combinations of cognitive measures. And here's the result of what's called the first canonical variate. So there are 8 graphs here correspond to the 8 brain states that we estimated, and the 4 bars within each refer to these temporal properties of fractional occupancy or mean lifetime, etc., of the state. And you can see that some of these frontal states are increasing generally in their occurrence, and some of the earlier visual areas are decreasing. And this pattern of increases and decreases related to a certain pattern of cognition. And this pattern seems to be dominated by fluid intelligence. So just to take my word for it, the dark orange bars would all be conventionally measures of fluid intelligence, while the lighter yellow, which are have less weighting overall, would tend to be more crystallized intelligence. And this is generally a decrease in fluid ability. The 2 bars that show an increase here are actually reaction time measures. So they do reflect worse performance because they're slower, where the ones that are negative are accuracy measures, which are generally worse. So this is a pattern of impaired cognitive performance associated with spending more time at rest in these sort of frontal parietal networks. And we can then ask, what about the effect of age? So if we plot this brain profile, the hidden Markov model profile on the left, against the cognitive profile up here, as a function of age, so young people in darker green and older people in lighter green, we see a significant increase in relationship in old age. So older people, their cognitive or their fluid intelligence at least appears to be more strongly related to this pattern of increased occupancy of frontal temporal states. So to summarize, the dynamics of brain states at rest do relate to fluid cognitive abilities, and this gets stronger in older age. In the paper, we speculate this particular pattern is more consistent with inefficient processing than functional compensation, because it's associated with cognitive decline, but you can read more in the paper if you're interested. And finally, in this section on functional connectivity, I want to talk about network segregation. So we've known with fMRI for many years that we can record functional activity at rest and estimate the correlation between time series of number of regions of interest. And if we look at the similarity of those, we tend to get some these large-scale brain networks, maybe 10 to 20 such networks that are strongly, the nodes within them are strongly intercorrelated, and they're less correlated with nodes in other networks. So functional segregation refers to how well separated these networks are. So it's, loosely speaking, the within network correlation or connectivity minus the between network connectivity. And so this is a paper from Chan et al. Using fMRI. And so they use a number of regions of interest and colors here. And if you project these onto 2-dimensional space, where the proximity in space is related to the strength of connections between them, you can see they form clusters of the blue network, the red network, and the purple network, which are more close with each other than they are, sorry, within each network than between. Or if you represent it a different way, if you have a connectivity matrix and order the ROIs by network, you tend to get higher connectivity within than between. And that measure segregation is simply the within connectivity minus between. So this is with fMRI, and these authors shown that, in fact, several papers have shown that this network segregation decreases as we get older. It may be an important property of networks breaking down in older age, which might contribute to cognitive decline. Indeed, they found, Chantal found correlations between changes in segregation and decreases in episodic memory. But what about, can you see this functional segregation with MEG? And this is current work ongoing, not published yet, by Ethan Knights. So we looked at the CamCAN data, and the first thing we did was replicate Chantal, that if you look at segregation on the y-axis against age, with fMRI, there's a quite reliable decrease with age, as others have found. But with MEG, we have the potential to look at multiple different frequency bands. So if you look at the prominent delta rhythm, which is 2 to 4 hertz, that showed a very modest decline with age, consistent with fMRI. Whereas if we look at the alpha rhythm, which is very prominent when you're resting, 8 to 12 hertz, this showed actually increased segregation with age. And then again, to the higher gamma, or at least low gamma, 30 to 48 hertz, we're now seeing a decrease again. So MEG is again revealing a much more complex picture. It isn't a uniform decrease in segregation networks. It depends on the frequency, which suggests that there may be different networks operating at different frequencies, some which decrease with age, some which increase with age. And we're currently rating that to cognition to see whether that's important, whether each of these frequency bands has important relationships with cognitive abilities. So at this interim stage, I wanted to summarize the first part to talk about healthy aging. And the main purpose again was to show that MEG goes beyond fMRI in studying the effects of age on functional activity, because there are many decreases in amplitude of the fMRI response to visual stimulation or auditory stimulation, which appear to affect vascular rather than neural effects. At the same time, MEG reveals, I showed you the work by Price et al. Showing that the latency of responses is recorded by MEG is affected by age, but these are shifts of tens of milliseconds, and that's well beyond the resolution of fMRI. Once you convolve those latency shifts with the canonical hemodynamic response function or the delayed vascular response, these would be undetectable. MEG also allows you to study functional connectivity, of course, and I showed you some brief evidence of brain microstates. Again, these are brain states that shift every 100 milliseconds or so, which is, again, beyond the resolution of fMRI. And these appear to relate to cognition, and they appear to be particularly important in older age. I also showed you that whereas we find a decrease in functional segregation with age with fMRI, with MEG we see a much more complex pattern of some decreases, some increases, depending on the frequency band, suggesting that these networks operate at different frequencies and have different properties. I've also only shown you a handful of studies from the main CamCAN authors. I just wanted to point out that there are many other groups around the world which who have downloaded the CamCAN MEG data and also made very interesting and important discoveries, and just list some of them here, showing that the community seems to value this large dataset of MEG data, which can also, because these are healthy people, can also function as a sort of normative data set if you want to compare MEG data from patients. And this leads me on to the second part of the talk about unhealthy aging or dementia. And I want to introduce now a different data set called the BioFIND data set. But before I do that, a bit of background. You've probably seen a graph like this before. So this is the hypothetical progression of dementia on the x-axis and normal to abnormal on the y-axis. And if you look at the brown line here, this is clinical function, so how well people cope in everyday life. And of course, when they surpass some criterion, we would give them, the clinician would give them a label of dementia. If we test more subtle cognitive tests, that's the purple line here, we find that earlier on, people are showing impairments of cognition. They're not so bad that it's affecting their everyday life, so they haven't been given diagnosis of dementia, but they're often said to have what's called mild cognitive impairment, or MCI, which is believed to be a prodromal state for Alzheimer's. So there's a period in time here where people fail some cognitive tests, but they still function reasonably well. But if we could detect this earlier, this may be helpful. And indeed, if you look at the other color lines, these are now brain measures. And it's been shown that measures like PET, that measure amyloid, this red line here, occurs often decades before cognitive impairment and dementia. The blue line here is structural MRI. So I showed you brain volumes at the start of this talk. And there may be a certain point in time when the neurons die because of a disease like Alzheimer's, and that shrinks the volume of the brain. That might be quite late on, relative to, for example, changes in neural function, particularly synaptic function, which you can measure with PET, but the idea here is you can also measure it with MEG. So the idea being that we can detect neurodegenerative diseases possibly earlier with measures like MEG than we can with standard structural MRI or cognitive tests. And that's the hypothesis which we can explore. And the BioFIND dataset allows you to do that. This was a collaborative work between colleagues in Madrid. Indeed, Fernando Mestu gave a MEG Masterclass a few months ago on their group's amazing work on dementia. So we pooled resources with them and also a group in Oxford. This is funded by the EU by a joint program in neurodegenerative disease, JPND. And we basically talked about how best to pool and harmonize our data. And the result of this a few years later was the BioFIND dataset, which, as far as I'm aware, is the first publicly available MEG dataset to study dementia. At the moment, it contains 324 participants, but we're constantly trying to grow it, who have at least 2 minutes of resting state MEG data. Roughly half are patients with mild cognitive impairment, and half of them are controls of similar age and male-female distribution. Half data are recorded in Cambridge, half Madrid, the same type of MEG machine, the old vector view, but different sites, which allows you to also look at generalizability of any MEG biomarker across sites, which will be important if it ever has clinical utility, because obviously every clinic will potentially have a different MEG machine, slightly different MEG data properties. The dataset also can, the vast majority also have a structural MRI, which you can use to help source localization, for example, of the MEG data. As I said, we hope to extend this with more. There are many ongoing projects in Cambridge and Madrid with MCI patients. Because the consent for these various studies was different to CamCAN, we can't let people copy the data directly. So instead, we use the Dementia Platform UK, DPUK, where a researcher can apply for access and then log onto their servers and analyze the data on the DPUK servers. Whether you want to use Linux or Windows, they can provide all the software you need. But the point is you can't take data away. So you can have to do analysis on their servers, but it's a very efficient platform. So if you were to go to the website, you'd find descriptions of the BioFIND dataset, and then you can follow instructions about how to get an account to analyze it. We actually published, well, we're in the process of publishing a protocol or data paper led by Delshad Vagheri. This is a preprint of the data paper, which describes in much more detail the nature of the BioFIND data. And we had some traveling brains and empty room recordings, etc., that are all useful. But another thing that Delshad did was ask, and this is now published last week, whether MEG provides complementary information in classifying MCI versus controls in the BioFIND dataset compared to the structural MRI. And for this, we use multi-kernel learning of support vector machines, again, to ask whether some MEG features, which we use sensor covariance, add information beyond structural MRI, or at least the gray matter volume in 110 ROIs, in terms of classifying MCI versus controls. So here is the, well, I'll show you in a moment. On the x-axis will be classification performance, where 50% is chance and 100% is perfect. By the way, you don't expect classification to ever be perfect, because there's obviously some errors in the clinical labels of MCI, which is difficult label to give. This is using tenfold cross-validation. And I'm going to show you a distribution of permutations of that different cross-validation to get some idea about the reliability of these estimates and accuracy. So here's performance of the MRI alone. On average, it was 71% correct. This is distribution across permutations. And that's very typical for MCI in the literature. If you look at MEG alone, it doesn't do as well as the MRI. It's only 68% accurate on average. There may actually be an advantage for MRI here in that the label of MCI that clinicians give the patients is often informed by a clinical MRI. So the MRI has a slight advantage in its classification potentially. But anyway, the more important point is that when we combine MEG with the MRI, using this multi-kernel learning, we now get up to 77% correct. And indeed, if we use the permutation tests, over 97% of the permutations result in better performance of the combined modalities compared to one modality or compared to MRI alone. And this demonstrates that MEG has complementary information beyond structural MRI in predicting mild cognitive impairment. This was actually most pronounced in higher frequencies, such as low gamma. Something we're doing at the moment, which is not published yet, is to see whether there's more information in the dynamics of these MEG resting state data. Again, using these hidden Markov models that I talked about earlier, rather than what we did in the previous paper I showed you, we used static covariance over the whole 8 minutes. But what if there's interesting information in the dynamics of these states? So again, using hidden Markov model, we use these various properties of mean lifetime, etc., as temporal properties, but we also added information about the transitions between states to see if there's a particular difference in MCI and controls in states that they tend to switch between. So there's additional set of features. So again, here's classification performance on the x-axis as a function of number of permutations. Here, we're with a static covariance, we're only 62% correct. This is obviously lower than the figure of 68 or whatever I presented in the previous slide. That's because we're actually using a much smaller set of ROIs to make the HMM estimable, but this potentially could increase. But the more important point is to ask whether the dynamic measures, such as the temporal properties, like the meaningful, etc., provide additional information, and there's not really strong increase. And the transition matrix was actually on its own worse than look at the static or dynamic covariance. Again, we can do this trick of saying, well, what if we combine together the static covariance, the temporal features, and the transition matrices? So we get up to 64%, which is an improvement on 85% of occasions. So it's not overwhelming, but it's a suggestion, at least, that the dynamics of these hidden states might be informative for MCI. But this further work's needed for this. And finally, I want to talk about what I think the future of MEG will be, at least for the study of aging and dementia, in terms of neurophysiological models. So a lot of people in the world are working on trying to bridge the gap between what we know about the dynamics and electrochemical properties of neurons through to what we can measure with MEG or MRI or fMRI. So we call this the microscale, and we know a lot about how the firing rate and action potentials relate to membrane potentials and how the membrane conductances change over time. These are reasonably well studied and can be expressed by a series of differential equations. But in between individual neurons, of course, when we measure MEG, we're averaging over millions of postsynaptic potentials or dendritic currents, all that happen to be aligned parallel to each other and perpendicular to the sensor. But we can sort of model this mesoscale by talking about populations of neurons with similar properties. So we have excitatory pyramidal cells in the superficial layer, for example. And now this kind of mass modeling level, we can talk about the properties of these populations and how they relate to each other and how they're connected to each other, and ultimately end up with networks of regions where the connectivity is determined by the temporal dynamics of this, are informed by what we know about typical neuron behavior. And ultimately, these systems, these coupled neuropopulations, can produce oscillations like the alpha and beta gamma that I mentioned that you can see with MEG. And they can also, if you ping them with a stimulus, can produce this typical damped oscillation that is an evoked response, an ERF with MEG. So they can produce things that look like evoked responses. And so James Rowe, who's a clinician neurologist in Cambridge, has been very interested in these models for a while to study the pharmacology of some neurodegenerative disorders, such as FTD or Alzheimer's. So we call this pharmaco-MEG. And this is a paper of James's led by Tali Adams. This uses a particular form of modeling called dynamic causal modeling. And so they use MEG data from an auditory mismatch task, a very common task, and they modeled 6 regions in left to right cortex of early auditory cortex, superior temporal gyrus, and inferior prefrontal cortex. And each of those regions itself contained a set of a population of neurons with different properties. So we had superficial pyramidal cells, deep pyramidal cells, spiny stellate cells, intermediate layers, and these kind of phasic neurons that needed for some of these drug effects that I'll talk about in a moment. So you have each region is itself a mixture of cell populations which are connected in certain ways, and we also connect between regions. And importantly, the parameters, the dynamics produced by these populations of cells can be related to neurotransmitters such as AMPA and GABA that we know about from neurophysiology. So they're getting a step closer to real brain biology. Again, these can, when you ping them with a stimulus, can produce these evoked responses that might differ for one tone from another. So the mismatch involves a series of tones, beep, 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 boop, and the boop is a mismatch. You look at the brain's response to that. And if you look at that, so the beep was the standard and the boop was what they call a deviant. What this study did by Adamstow in Journal Neuroscience is take healthy people and give them a GABAergic drug, and then look at the effect of that on the parameters of the model when the model's been fit to the MEG data. And I won't go through the details, but this is a placebo group and the drug group, Tiger Bean. And there are some parameters of the models where there was an significant effect of the drug on the physiological parameters, which made physiological sense. So now we know that these models can capture something about a drug, and we can also then take this to patients who might have a deficit in certain neurotransmitters and see if we can measure with MEG properties of the neurons and possibly how that might be normalized with a drug. So I think this is an exciting area for MEGs to use the dynamics of MEG to try and inform the neurophysiological modeling, which includes realistic assumptions about populations of neurons with different dynamic properties. So I'll finish there, but just wanted to say a big thanks to all the participants who took part in CamCAN and in the BioFIND dataset. Our funders, primarily MRC, the BBSRC, who funded CamCAN, and the EU. The people where I work at the MRC CBU, all these people listed here who have helped with CamCAN over the last 10 years, and people here in Madrid and Oxford who've helped with the BioFIND dataset. So thank you very much. And this is just a outline summary of what I said if you want to ask any questions.
Hi, so we are now entering the Q&A portion again of this webinar. And so the first question that we have for Professor Henson is, what is the difference between metastates and microstates? Can you please explain them in relation to MRI and MEG?
Sure, I'm not sure what the questioner means by metastates. But yes, so microstates was a phrase I used, has actually come from EEG research, where they did try and, for many years, separate the brain dynamics into stable states using sort of clustering techniques, looking for stable spatial topographies over the electrodes. And then HMM modeling is a more recent application, and and has been applied to MEG data, but could also be applied to EEG data. And there, so the word microstate was borrowed from that literature of EEG microstates. I don't think it has a particular definition or meaning relative to metastates in this context of dynamic model, but maybe I don't know. So happy to follow up offline if anyone wants to explain to me the proper definition of micro and meta for this context. But then there's this idea of microscales, mesoscales, and macroscales I alluded to in this last part of my talk in neurophysiological modeling, where yes, you have the microscale refers to what we know about the behavior of neurons. We know a lot about how the neural electrical properties change and how action potentials work, but we have to bridge that to the mesoscale, which is now populations of thousands of neurons, and we can approximate the behavior of a large number of neurons using methods from statistical mechanics and physics, so-called neural mass models. So we can kind of abstract away from individual neurons or particles into the behavior of a large population. And at that level, we might be able to start making contact with brain regions and electrical activity in those regions estimated from the macro scale, which is the MEG sensors or the fMRI machine. So that's the concept of the different scales in terms of the neurophysical modeling. But if the questioner has a particular definition of metastates, then I'm very happy to pursue that offline.
Okay, so next question is it necessary to localize the sources of MEG if only used for classification of participants?
Yeah, that's a good point in the sense that most source inversion techniques are linear operators, so you don't gain more information if you want to classify individuals by going to source space, apart from the fact that you realign the sources better by having a head model that takes into account the position of the head relative to the sensors. But the issue comes when you start using second order statistics, like in the data I showed, we use the covariance matrix. And the covariance of the sensors is not a linear relationship with the covariance of the sources, in which case it is better to try and estimate the sources if you want to use these second order statistics, because the linear relationship between sensors and sources is now broken. And there are other techniques. There's Riemannian manifold embedding, which you can do to transform sensor level covariance matrices into which respects the geometry of that, to then do classification. And people have shown that that performs as well as source localization in many cases. So you don't need to do source localization if you're just interested in classification. If you're interested in covariance matrices and use a Riemannian projection, and that means you don't have to have an MRI or all the problems associated with source localization, which is obviously, as people know, an ill-posed problem with no unique solution. But yes, I think there are more sophisticated ways to deal with keep data in sensor space if you're only interested in classification of individuals into two groups or predict age or something.
Okay, so next question is, do you have MEG data from people with genetic risk of Alzheimer's but no symptoms yet?
Not really. I mean, there are large cohorts out there of individuals who have genetic risk or familial risk of Alzheimer's who have been studied with MRI or other brain imaging techniques. I don't think, I might be wrong, I'm not aware of at least a publicly available data set with MEG. What we have got in CamCAN, which I didn't talk about, was a subgroup of people who performed below the normal cutoff of the mini mental state examination for mild cognitive impairment, yet had no symptoms and had not been to their clinician to complain of any problems. So these could be a pre-symptomatic group. We are following them up, or they could just be had a bad day on the test. I mean, we've obviously repeating the mini mental, and some people consistently perform low, but we need to know whether they actually have any neurodegeneration in their brains, and that's the CamCAN frail cohort, which we're currently following up. But I didn't talk about it today. But it's a very interesting question about if we can detect signs in brain activity very early, even before symptoms, so even before MCI. And again, Fernando Metsu, who spoke at a masterclass a few months ago from Madrid, has also got a cohort of these people, and he's studying them with MEG. So yeah, so that's an interesting active area of research.
So what does MEG offer beyond EEG?
Yeah, good question. I did mention that at the start. Obviously, MEG is a more expensive technology, and it is yet to appear in every hospital. But what it does offer is improved spatial resolution. So it's still obviously not the spatial resolution of fMRI or PET, because it's essentially a topographic rather than tomographic technique. So we know with MEG we can't localize all parts of the brain very accurately. Some parts we can localize well on the superficial part of the brain, but deep brain regions are hard to localize confidently with MEG. But nonetheless, it still offers better spatial resolution than EEG. And the reason for that is the basic physics or Maxwell's equations that tell us how magnetic and electrical fields are affected by different conductivities of tissue within the skull. The skull itself, the CSF and brain tissue have different electrical conductivities. That's very important to get right for modeling EEG, but it's not so important for MEG. And that's why you can localize a bit more confidence and you have more spatial degrees of freedom, typically with a MEG dataset than an EEG dataset. And that means that if you're doing anything to graph theory or modeling networks, you can potentially resolve more nodes, even if you can't resolve as many nodes as you can with fMRI. You can resolve more nodes with MEG than you can with EEG. So you may be more likely to see effects of Alzheimer's, for example, on properties like segregation or some network property. That's potentially the hope of MEG. In reality, we often record both at the same time and use both because they are somewhat complementary.
Are there ethical issues about sharing brain data?
Yes. So there's been a kind of transition in the field over the last 10 years, with GDPR, which is a European law about data sharing. So it used to be thought that data was the property of the researchers or the funders, and GDPR makes it very clear that data are property of the individuals who provide the data, the volunteers themselves. And if they give consent to sharing, then you can share the data, as long as it's informed consent, as long as they understand the risks. Now, most stuff we share on CamCAN is reaction times and accuracy, and it's not easy. You can't identify someone very easily from those data. But things like a brain scan, a T1 weighted MRI scan, is potentially identifiable because it contains, we all have quite distinct gyral and sulcal patterns like fingerprints. So if you had an MRI already, you could potentially decode information and then retrieve other information about the same person from a database. So you do have to be wary of sharing brain data if it can be linked to an individual. But as long as you have consent from that individual that you can share and they understand the risk of that, then it's fine to do so. Well, at least that's my interpretation of GDPR. So you do have to be careful. You have to get consent. And there's more and more realization in the scientific field that sharing data is a good thing to improve reproducibility, to increase sample sizes, to do larger meta-analysis and mega-analysis. So it's a good thing generally to share data in science. And we just need to be, when we collect the data, we need to be aware up front that we ask people for that permission to share data.
And the final question is, are there genetic components to MEG data?
I think there are. I think there's a paper from the Oxford group, maybe Kolkoff et al. Looked, I think, at twins, mono and dizygotic twins, and I think there are, if you look at the mechanism, there are strong genetic influences. We haven't looked at that in CamCAN. I think our sample size is probably a bit small. 700 people is not typically enough to, at least for gene-wise association studies or GWAS studies. But as the amount of data available from MEG data increases, maybe we'll be able to look for genetic components to brain oscillations. I'm sure there are, and particularly some genes that might be risk factors for Alzheimer's, for example, like or ApoE, might be good candidates to look for. We have got saliva samples in CamCAN. We have done some analysis of SNPs, common SNPs, and we didn't find a relationship between ApoE and MEG. But again, I think that's because our sample size is too small. But yeah, that's where the benefit of pooling data from across sites and internationally can give us sample sizes that you need to do proper genetic analysis of MEG data, and which has potential benefits for neurodegeneration, etc., if we can find genes associated with those diseases.
Well, thank you, Professor Henson, for your time today. This has been an excellent session. And thank you, everybody, for attending. And we hope that you'll join us for future MEG and Masterclasses. Thanks, everyone.
Bye.
Bye.